Our next panelist is Ben Dichter. He is a research software engineer, founder of Catalyst Neuro, and also the community liaison for Neurodata Without Borders.
All right, hi everyone. I'm Ben Dichter, and I'll be talking to you about how we are trying to drive collaboration in neuroscience through open data and software. I'm the founder of Catalyst Neuro. So let me just talk first briefly about the need for open data in neurophysiology. So the data that we collect is very valuable and precious. It can take lots of money to collect the equipment. It can take lots of time, both in terms of person hours of highly trained people, as well as months or years of time to collect the data. And we use animal lives. Often we end up sacrificing the animals that we use. So it's really important that we derive as much value as we can from this data. Through listening to these talks, I've noticed that there's actually another way in which this is valuable, in that it can be quite scarce. A lot of the talks in the last 2 days have involved field research, which collect data that may be impossible to collect ever again. And by its very nature, a lot of this data is really impossible to collect again because it involves changing ecosystems. So it's really important that we document this data because many times it can be used to answer many different scientific questions. There have been several talks also that talked about, to answer this question, we look back at data we had already collected in our lab. And that's just a demonstration of the fact that this data is highly valuable, can be used to answer many questions, not just within your lab, but outside your lab. The problem is that sharing data is very tedious. It can take months of back and forth communication if the person who collected the data is still in the lab. If they're not in the lab, it might be hopeless. And so as a result, it's often more convenient just to collect new data and write new software every time. We think this is an enormous waste, and so this is a problem that we're trying to fix in the field. We think this is one of the biggest impediments to the progress of neuroscience, and this is what we've chosen to tackle. And when I say we, I mean Catalyst Neuro. So I'm the founder of a small consulting company called Catalyst Neuro. We're a team of research software engineers that are both software engineers and neuroscientists. And we connect data producers, tool builders, and data users in neurophysiology research. So the main tools that we used for this are Neurodata Without Borders and Dandy. So I should say the Kavli Foundation started NWB about 10 years ago. And this is an effort led by Oliver Rubel at Lawrence Berkeley National Lab, which I'm a partner with him on. And this is a data standard and software ecosystem for storing and analyzing neurophysiology and behavioral data. And then there's also the Dandy Archive, which is led by Satra and Yarrick. And this is an archive and collaboration space for sharing and analyzing neurophysiology data. And so if you're wondering what's the difference between these two things, I like to think of them as NWB is kind of like MP4. It can be a data standard for how to represent the data so that you can build software around it and share it with your colleagues. And then Dandy is kind of like YouTube. It provides a place where you can upload standardized data, you can download it, you can search for it and stream it all for free. So NWB, let's dig in a little bit to what NWB actually is. It's a format and a data standard with the goal of packaging all of the data with the metadata necessary for reanalysis. So this includes the neural activity, of course, the actual raw recordings. It also includes metadata about data acquisition, experiment design, subjects, the behavior of those subjects. And crucially, it also provides the ability to create extensions so you can expand the format to handle new types of acquisition systems or analysis methods or lab-specific metadata. So how do we actually get people from where they currently are, which is this huge diverse zoo of different data standards and data formats, into something standardized? Well, one way you could do it is by sticks, such as funding ultimatums, and we've already seen these, and they are a powerful motivator to do something, but not necessarily to use the best data curation practices. So on their own, they're not that effective. Carrots, such as analysis and visualization tools, are really great. If you can provide people with tools that actually accelerate their research, they'll be motivated to make this transition into changing their data formats and building around it. contributing to that ecosystem. A lot of what we focus on is actually guidance. It's actually lowering this energy barrier directly by documentation, tutorials, hackathons, working directly with neuroscience labs, building and distributing open source tools to facilitate this transition. So I'll just show you a big snapshot of, we work with dozens of different analysis and visualization tools, state-of-the-art tools for data processing, visualization, data management across different types of neurophysiology data modalities and different programming languages. And we also work on building tools to facilitate conversion of this data into NWB. The most recent one, which is the most automated, is called NWB Guide, which stands for GUI, or Graphical User Interface for Data Entry. And this, I should say, was funded by the Kali Foundation. And we kind of think of it as like a TurboTax for converting your data, where you just tell us where your data is, we'll extract as much metadata as we can can read over 40 different common neurophysiology data formats, and then we can automatically convert that to NWB, automatically packaging the data optimized for archival storage and cloud compute, and even help you upload automatically that data to Dandy if it's ready to be shared with the community. Then there's NeuroConv, which is a low-code solution, so you can convert data with about 8 to 10 lines of Python code where it would be 100 lines of code beforehand. And then if you really want to go into the nitty-gritty, there's PyNWB and MatNWB, where you'd be responsible for loading the data from your various data formats, but you have the most control about how that would be input into NWB. So once you convert your data, you can deposit it to the Dandy archive. So Dandy stores neurophysiology data and associated data in the NWB format, which allows it to validate NWB, check for best practices, automatically extract metadata that enables search over all of these different assets. You can upload hundreds of terabytes of data to Dandy for free, which is pretty wild, and it's made possible because of a partnership we have with Amazon. So AWS Open Data Sponsorship Program allows us to upload data to their free S3 buckets for free, download for free, store it for free. We've been doing this for years. We have hundreds of terabytes of data. It's working great. It can handle the raw data from most neurophysiology experiments. We just ask if you talk to us if your data set is over 10 terabytes, just to make sure that it's like packaged in a way where it's maximally reusable, but we welcome data sets over 10 terabytes. And I should say that you can upload data in public mode or in embargo mode where only you and your co-owners have access to this data until you're ready to unembargo the data. So at Catalyst Neuro, one of the big things we've been doing is engaging with individual labs, helping them build custom conversion pipelines and upload their data to Dandy. To date, we've worked with over 50 labs, helping them build custom conversion pipelines. And all of these conversion pipelines are open source on our GitHub organization. So with all of this work, I'm very proud to say that at this point, we have over 350 open neurophysiology data sets on the Dandy archive. Actually, most of this is contributed by the community, not by work that we've done specifically. Also, you'll see this super linear growth where there was just after the NCE conference #1 in November of 2022, you see this inflection point after which we have skyrocketed contributions to this archive. So I don't know if there's a causal relationship, but I just thought it'd be interesting to point out. I know there's a lot of interest in diverse species here, so I wanted to make sure to point this out. Definitely our most common species are the ones you would expect: mice, rats, humans, monkeys, zebrafish, fruit flies. But we welcome any species, and we have lots of different types of species. We've got an ant data set, and we've got crayfish. There are a lot of species mentioned here that we don't have yet, but we really welcome data sets from those species as well. We have extracellular electrophysiology data. That's the most common type of data set that we have. We also have a good number of calcium imaging and intracellular electrophysiology data sets. I want to mention, especially since we're here at the Allen Institute, that some of our most reusable, highest value, really exciting data sets are from the Allen Institute. We have 10 data sets from the OpenScope program that involve 2P calcium imaging data, neuropictal electrophysiology data doing different visual tasks. From the BICAN initiative, there are 10 data sets that store the electrophysiology of patch-seq recordings. So this speaks to integration across different types of modalities, because the patch-seq also has genetic information in other repositories. And then going back to the Brain Observatory, where these really high-yield data sets, where Allen Institute really pioneered this. We have some data from that as well, which Saskia helped us put together. If you're interested in analyzing open data, I highly recommend checking out these Allen Institute data sets. They're really great data sets. So we're seeing data reuse. I mean, this whole effort is kind of pointless if people don't actually go in and use the data. So we're starting to track data reuse, and we've identified 30 papers where people use NWB data on the Dandy archive. These include high-profile papers like the Nature paper about the zebra algorithm. This figure from that paper was actually derived from an NWB data set on Dandy. And we're also starting to see new, really interesting techniques, like people using these open data sets to train transformer models of neural activity. So we're really excited about these different use cases, and we want to facilitate different types of use cases and analyses of these open data sets. So the question is, how can we facilitate that? One way is to integrate state-of-the-art tools, to just continue to engage with the community and have better and better data exploration, visualization, analysis, partner with compute resources. We can provide better training, conferences, workshops, invited talks. We haven't done much in terms of building course material that could be integrated into undergrad courses, but I'd really like to work on that if anyone's interested. And we're really deep into looking at how we can facilitate this with artificial intelligence, using large language models to curate data sets, to find relevant data sets for a given scientific question, and even to write analysis code that can teach people how to dig into these data sets. I want to thank our funders, in particular, of course, the Kavli Foundation, also Simon's Foundation, Michael J. Fox, and Brain Initiative have been generous supporters of this work. This is a huge interdisciplinary team. I just wanted to quickly show you actually how you would go on the archive, because it's really a lot simpler than you would think. You can just go to dandyarchive.org. I'm gonna look up a data set from the Moser lab. This was one of the data sets that was cited in their Nobel Prize. You see a DandySet landing page here. You can go into files. I'm just gonna go to the first session of the first subject. You can download it right here. It's only 14 megabytes, but I'm gonna open it with a program called NeuroSift. This streams data directly from the archive into your browser and visualizes it in real time. So I'm gonna visualize the electrical series, a raster plot, and the spatial information as a time series and as X versus Y. And I can just click this, and this data is streamed in. And you don't even need a Dandy account to do this. Any of you could literally go on here and do this. Well, this is internet dependent, so we'll see. But it's starting to stream in. But this will show the raw electrical series. This will show spike times of 8 sorted units. I'm going to increase this to actually show 8. This shows X and Y position over time, and this shows X versus Y. So you can really start to dig in without doing any programming, but you probably will at some point want to do programming. So the way you would do that, you can scroll down here to Python usage, and this shows you exactly the code you would need to stream this data into a Python session. So I'm going to copy this and put it into this notebook and execute it, and then I'm going to show you how you would analyze it. So I'm going to import pineapple as nap, import matplotlib.pyplot as plt. Nope, there's too many P's. Okay, nap.nwb file. Okay, so this will open the NWB file in a tool called Pineapple, and this exposes the data streams that you can analyze here. So I'll do nap.compute 2D tuning curve, NWB file. I want the units and the spatial series LED1 with 20 bins. So this will actually do maps.bins. This will actually compute the rate maps for the individual units. So this was a data set used to derive the grid cells found in the entorhinal cortex. So I'm going to see if I can reproduce that. So while that's running, I'm just going to write the plotting code. So that's for x in maps.values plt.im show x, and then I will plt.figure. And then while this is running, color bar, oh, it's already done. Forget the color bar. All right, so there you go. Grid cells derived from the Moser Lab data sets streamed directly from the archive. So, Nobel Prize speed run. Yeah, but this, I hope, shows off the potential of these data sets. This is one out of over 350 data sets, and I would really love to work with any of you to share your data or to use the existing data sets on the archive. Thanks.
So we have time for one or two questions. Oh, Ben, you got a question.
Yeah, technical question. So unless I misunderstood something, so you can standardize, the goal is to standardize data in a format that it kind of every person can open it and look at it, right? So how do you deal with specific softwares, like if you do imaging on the Zeiss, it's Zen.
Yep.
If you do imaging on Leica, they have their own. If
Yeah.
you work on Olympus, they have
100%.
their- So how do you deal with
So
it?
we've worked with dozens of different labs, and we've been exposed to dozens of different data formats. So that application that I showed you, that I mentioned, the guide and NeuroConv, they know how to read the data and metadata from over 45 different neurophysiology data formats, including, your Zeiss and your Thor labs and your different microscope systems, over a dozen different electrophysiology systems, but also the common analysis pipeline. So if you run Kilosort and Phi on your electrophysiology, or if you run Sweet2P on your calcium imaging, we've just built ways to ingest. We've just meticulously gone through and built data importers for a lot of the most popular ones. Now, I want to say all the most popular ones, but we just keep on seeing more and more. I mean, it just never ends. But at least these days, most of the time that we engage with labs, we have already built a tool that works with their data.
Do you work with companies like Zeiss or Leica?
Yeah,
Or-
sometimes
and tell,
we-
like, hey guys, maybe we should just, maybe you, three of you, like, just go to the same format. Then you don't need to convert it.
Yeah, I mean, so I should say there is another effort that was also funded by the Kavli Foundation to build a C++ library that could be plugged into these acquisition systems so that they can write directly to NWB. The reception of that, some companies have been really excited about it, some not so much. They want really good write performance, and so they kind of are optimized for that. That's kind of the optimistic view. The other side is some of them have like analysis ecosystems that they also sell, so they're not so excited about directly exporting to NWB. But we do work with the companies to at least have the state-of-the-art tools for reading the data and metadata so that we can do the best conversion possible into NWB post-hoc if that's what we need to do.
Thank you.
Okay, thank you.